So I'm going to talk about some science today. And I'd like to start by sharing some statistics that kind of inform and drive the research we do at Riley Children's Hospital and the Wells Center for Pediatric Research. And that is that in 1950, if a child was born with a severe congenital heart defect, in other words, a birth defect that affects the function of the heart, in 1950, that kid had a little less than a 20% chance of surviving their first year of life. Nowadays, due to advances in cardiothoracic medicine and other forms of specialized medical care, we're up to about a 95% survival rate. However, these kids aren't out of the woods, because if you're born with a severe congenital heart defect, and about 1% of the population that's 1 out of every 100 live births. If you're born with severe congenital heart defects, pediatric surgeons can fix this, but they can't cure it. So you're at an increased risk for the rest of your life from suffering from, for example, a life-threatening arrhythmia. And so when you take into consideration severe heart defects that are evident at birth and couple those with birth defects that may not become evident until you're in your 60s, it's estimated that about a little less than 500,000 children and about 1 million adults are living with a congenital heart defect in the US. So I study how genes are turned on and off in the cells of a developing embryo, because every cell in your body expresses a specific profile of genes that imparts a unique function to those cells. So in other words, in the heart, there's a population of cells that are known as the conduction system. And these cells are special because they generate and propagate electrical signals. And these electrical signals tell the heart to contract. They control the heartbeat. When the conduction system doesn't develop properly, a patient can experience a life-threatening arrhythmia. That is an abnormal heartbeat, and they're at an increased risk for sudden cardiac death. So how do we tell when the conduction system is dysfunctional? We diagnose this using an ECG, which you've heard a little bit about today. As medical procedures go, ECGs are relatively inexpensive and easy to do. And because of this, you can collect data from a large number of patients. Additionally, because it's the future, we can sequence whole genomes relatively inexpensively and easily. And so we can compare small changes in the genome, in the A's, the T's, the C's, and the G's that make up our DNA, with the likelihood of having an abnormal heartbeat. And because of this, we can identify specific genes that are associated with abnormal heart rhythm. And one of these genes happens to be one of the genes I study, called HAND1. And so we're going to go into the science. So this is just a stick figure representing, well, the mouse is being a little uncooperative. Oh, there we go. This is a stick figure representing a segment of DNA. And the HAND1 gene is over here on the right. So this actually makes the HAND1 protein that your cells need to function. But the variations that are associated with an abnormal heartbeat don't occur within this region. They actually, Anyway, they occur in the neighborhood, in a region that it's not really clear what this DNA is supposed to be doing. But because of some markers, we reasoned that potentially this is a switch that is important for turning HAND1 on and off. And so how do we test this? Because you can't actually just look at cells and say, oh, this gene's on, this gene's off. So we used some nice genetic engineering. We isolated this region and hooked it up to a bacterial gene that you can stain blue. And we made some genetically engineered mice. And my hope is that you can see that the blue staining is this is a mouse embryo, the blue staining is in the heart. So we have a genetic switch that can turn on a gene in the heart. And so what's regulating this switch? Well, there are some sequences. So in this switch, it's not really doing anything in and of itself. It's basically a docking platform for these proteins called transcription factors, and they do the heavy lifting. They turn genes on and off. And there are specialized transcription factors called GATA factors. And they're called GATA factors because they bind to the sequence GATA, because scientists are literal. And so we found 2 GATA sites in this switch. And if we go back to our genetically engineered construct and we mutate these GATA sites so GATA factors can't bind to them anymore, and we make more genetically engineered mice, there's no more expression in the heart. There's no blue staining there. So we know that GATA factors bind to the switch and turn it on. But what does this switch actually do? We used a relatively new genetic tool called CRISPR-Cas9. You may have heard of it. It's brand new, and it's a really efficient and effective way to modify the genome. We use this CRISPR-Cas9 to just delete the switch. So what happens when we delete the switch? This is another method of visualizing gene expression in a developing embryo, and I hope you can see that this purple stain represents where hand 1 is turned on. In these mutants where we've deleted the switch, there's no more purple staining. Hand 1's gone. And so what happens in these mice? When we give them an ECG, they have an abnormal heartbeat. Their conduction system is dysfunctional. So what does this mean for human patients? Well, like I said, we were looking at how tiny variations, single base pair changes, can have an effect on the function of the heart. And we saw that there's an A within the second GATA site that in certain patients with arrhythmias, this A is switched to a G. And I'm not going to get into the detail of it because it's a lot of hardcore data, but basically, in the patients that have this G, GATA factors don't bind as well. So we can envision a scenario where GATA factors don't bind as effectively to the patients that have this single base pair change, and because of that, they don't make enough hand 1, and because they don't make enough hand 1, their conduction system doesn't develop properly, and because of that, they experience arrhythmias. And so we're at the cusp of a sea change in personalized medicine, where potentially in the future, this CRISPR-Cas9 might be able to change these single base pair variations to a non-life-threatening condition. And it gives me hope, because I think I really am excited to see in 10 years what the new statistics are going to be and how far we've come. So thank you very much, guys.